Hello, everyone. My name is Martin Hoenigl. I'm associate professor at the Medical University of Graz, Division of Infectious Diseases, and also have an appointment at the University of California, San Diego. Today, my job is to convince you that CAPA has actually been overemphasized in the literature. These are my disclosures. So you can see that I really have a hard job today because I'm working against this particular guy that you see on screen here, who is emphasizing since 18 months or so now that CAPA is a very important disease and COVID in a significant number of patients. But I will prove today that this guy is wrong and actually CAPA has been totally overemphasized. So if we look to the incidence of CAPA and other mold infections in the early phases of the pandemic, we can see that there have been tremendously high rates reported. We see here in ICU patients a median of 8.9%. In all ICU patients with COVID-19. In patients with mechanical ventilation, this reached up over 20%, 20.1% in median. With incidence rates in some of these studies up to 38%. So nearly 1 out of 2 patients developed COVID-associated pulmonary aspergillosis, which of course is hard to believe. But when we really look into the details of this study, and because it was the first study, and I really want to congratulate authors for moving this study along and raising the awareness for CAPA. So this was a French study by Alexandre Alagneau and colleagues where they found 33% of their relatively small group of 27 mechanically ventilated COVID-19 patients developed putative or proven CAPA. But when we look into details there, it becomes very clear that not all these putative diagnoses are strong diagnoses. And questions arise, obviously, from there. For example, how to interpret positive BL culture when BL galactomannan at the same time is negative, which was the case in a number of these patients. You see all the blue ones. What should you use as BL galactomannan cutoff? Is 1.0 ODI not the optimal cutoff in non-neutropenic patients, or is it fine to use a lower cutoff? And also, what to do with patients where a slightly elevated serum galactomannan is the only mycological evidence for COVID-associated pulmonary aspergillosis. And then last but not least, what does beta-D-glucan mean in intensive care unit patients with COVID-19? Is it really a marker for aspergillosis, or is it rather a marker of a leaky gut, a marker of sepsis that correlates with SOFA scores, and really has nothing to do per se with aspergillosis itself, but really a marker, overall prognostic marker, but not indicating aspergillosis. How do we look in autopsy studies? And there is a large review of many small autopsy studies and case series summarizing 50 studies where 677 dissidents were included. 155 of those, although only minimal invasive autopsy, but that still leaves 522 dissidents dying of COVID-19 who received a full autopsy. And of those, only 2.1% had actually invasive mold disease at autopsy, which is 11. We have now made, of course, some advances. So early on, we did not have clear criteria on how to define CAPA. And we used definitely very weak mycological evidence like tracheal secretion culture, serum beta-D-glucan, as I showed you before, to define COVID-associated pulmonary aspergillosis. Now we do have consensus criteria, and at least they work to some extent that they bring down the actual rates of CAPA reported in studies from 20% in mechanically ventilated patients to around 10% in mechanically ventilated patients. So we're getting a little bit closer to the 2% reported in these autopsy studies. And this has been also shown in large multi-center studies. For example, here, mainly European multi-center study where the median incidence was 10.7%, so exactly in this range. Again, however, still a wide range between the participating centers, also in this study, with the new criteria. Same findings also with the other large multi-center studies, between 10% and 15%. The prevalence using these new criteria in mechanically ventilated COVID-19 patients. However, of course, the criteria came out at a time where we did not have strong evidence for some of the diagnostic tests recommended there. And while there were significant advancement versus what we had before in the absence of criteria, there's still some question marks also with some of the tests that are included here. One of them is, for example, the lateral flow assay, which works pretty well and is well evaluated in bronchoalveolar lavage, but there's much less data for this assay in serum. And what has been shown here by these multi-center studies between Graz, Rennes, and Cardiff was that especially if the serum lateral flow assay is the only evidence for CAPA, this may really be not true evidence. So we had 7 reclassifications from no COVID-associated pulmonary aspergillosis to probable COVID-associated pulmonary aspergillosis just because a slightly positive lateral flow assay. And this is, of course, key because also with the new criteria, we are still in an area where we don't have clinical evidence, we don't have radiological evidence. The only thing that guides us for CAPA diagnosis is the mycological evidence. So these things are key in order to avoid overestimation. And of course, the same problem, as I said before, probably also exists with serum galactomannan, where maybe a single slightly elevated serum galactomannan may not be considered sufficient evidence for CAPA if it's a standalone result and everything else is negative. So still some work to do there. And of course, this work will be done. Nevertheless, I have to emphasize how important these criteria are for classifying CAPA and how much advancement they brought us. How do we look now for prognostic potential of diagnostic tests for COVID-associated pulmonary aspergillosis? And there have been 2 recent studies. And the they both found is basically that the one major predictor for mortality in patients with CAPA is angioinvasion, and is as a sign of angioinvasion a positive serum galactomannan test. So this is the major predictor of mortality. Also, of course, beta-D-glucan, positive serum beta-D-glucan is predicting mortality, but probably not mortality due to CAPA, but it's a general mortality predictor as it correlates very strong with SOFA scores in COVID-19 patients. And there's now also been a second study, again from the same French group that I showed before, that published the first study on CAPA in their ICU from Alan Yu and colleagues, here led by Sarah Delier and colleagues, where they also looked into 176 ICU COVID-19 cases with a significant amount of about 24% probable and also some possible cases of CAPA. And again, what they found is that the major predictor of mortality was either a positive serum galactomannan or plasma Aspergillus PCR or a positive direct examination of BL. So these were predictive of mortality, and unfortunately, these were so strongly predictive that all of these patients who had one of these died, independent of antifungal. 100% in patients who had one of these test results positive. And that also, of course, has important indications in terms of when to diagnose CAPA and when to start treating CAPA. Because obviously, when we diagnose this at a point where everybody dies anyways, we can potentially, and this is, of course, a little bit controversial, but potentially I could say we can save antifungal therapy in these patients because they would have a fatal outcome anyways. Of course, we're not doing that. This is just for the debate. But of course, the goal is not to diagnose it late. That means it's really vital to diagnose CAPA in the early phases when it's still only airway invasive. And this is taking quite some while in COVID-associated pulmonary aspergillosis, up to 5-7 days, where it's only growing airway invasive and not yet being angio invasive. And we only have positive markers in BL, but not in serum. So it's vital to diagnose at this early stage in order to save our patients. And then, of course, when we diagnose early, we also need to treat early because diagnosis without treatment does not help. And there's also now clear recommendations about what to use for treatment. Voriconazole, isavuconazole first line, liposomal amphotericin B second line. And this is important because nevertheless, I told you it's probably overemphasized. Not all of these patients that are reported in the literature have angio-invasive CAPA. And we don't know if everyone who has airway invasive CAPA would advance to angio-invasive CAPA. But nevertheless, we also know from literature that these patients who also have airway invasive, even only have airway invasive CAPA, have a worse outcome. And CAPA is an independent predictor of ICU survival, and a negative predictor of ICU survival in these patients, also if it's only airway invasive. And that's why certain centers, and of course, I'm arguing against these centers. Unfortunately, one of them is my center, where we now use routinely prophylaxis against CAPA in our patients, because we saw the significant decrease in CAPA incidence from 17.5% to 1.4% in our study. So it's now part of our standard operating procedure to use posaconazole prophylaxis in mechanically ventilated ICU patients with COVID-19. And that really brings me to my conclusion, and I'm happy to open up the stage for Michele, who will take the point against me. But I really want to emphasize here that I think, especially in the early phases, CAPA rates are overestimated, especially in the absence of reliable consensus criteria, and all sorts of, unspecific mycological evidence was used to classify CAPA, including positive tracheal secretion, sometimes even positive sputum, or serum beta-D-glucan levels. The ECMM-ISHAM criteria have been a breakthrough, but it's of course a breakthrough that still needs constant updates. And I think, they really have worked well to bring us down close to what we're seeing in autopsies. But nevertheless, I think we still need to work on some of the details, including whether a single serum galactomannan or slightly elevated lateral flow assay above 0.5 or between 0.5 and 0.6 is really enough evidence for COVID-associated pulmonary aspergillosis, and also what to do if results are going the different directions. For example, we have a positive culture, but everything PL, including galactomannan, is negative. How to interpret these results? That all said, it's vital to diagnose and treat CAPA early, while it's still only very invasive. And there's really no indication to wait until your serum galactomannan becomes positive, but at this time point, because at this time point it's too late and your patient will die anyways. So you need to go into the lung, go where the money is, and diagnose early and treat early. And last but not least, of course, when thinking about prophylaxis, this is also something that needs to be evaluated not only in single center epidemiological small cohort studies, but really in randomized controlled trials to get some more evidence. And with that, I want to thank you for your attention.